Hello, everyone. Thank you for joining us at this session dedicated to the role of European reference networks in the clinical trials in epilepsy. My name is Katarzyna Kotulska from Poland, and together with my friend Valentina De Georgis, we'll be chairing this, I think, very important and informative session. I will give you a brief overview of the European reference networks, what they are, and I think it doesn't work. Okay, here it is. So the European reference networks were...
You have to click just one time, I think.
Yeah.
Here.
I tried it. Okay. So the European reference networks are groups of hospitals launched in 2017 by the European Commission for the treatment of the rare and complex disorders to equalize the care of the patients with rare diseases with the so-called common disorders. As you can see, epilepsy was there from the very beginning. You can see it's on the picture showing Helen Cross, the former coordinator of European reference network dedicated to rare and complex epilepsies called EpiCARE during the very first meeting of ERNs in Vilnius in 2017. This year, the European reference networks had the results of the second call for the memberships in the network. So currently, in the 24 networks, we have nearly over 1,500 hospitals and centers included in the networks in 382 hospitals across the countries of member states of European Union and Norway. So the landscape of functioning of European reference networks is very complex. One of the roles of ERNs is to not only do research, but also to work on the future potential therapeutic possibilities in rare and complex disorders. So we have to collaborate with different entities, including regulators, including the founders, including the pharma companies, and of course the researchers. And this is what our presenters will be giving talks about, I think. So I'm ending up with this, and I would like to ask Valentina to introduce our first speaker.
Yes, thank you. And even if it could be pleonastic, it is my honor to introduce you Professor Emilio Perucca, neurologist and clinical pharmacologist, past president of the International League Against Epilepsy and Italian League Against Epilepsy, currently clinical professor at Melbourne Hospital, Australia. To me, a mentor, and most important, one of the foremost experts in clinical epilepsy clinical trials and pharmacology of anti-seizure medication. In this session, Professor Perucca will discuss a critical review of the recent EMA guidelines.
Well, thank you, Valentina, and thank you, Kasia, and good afternoon to everyone. Valentina, you managed to embarrass me, even though it went far above what was expected. So it's a bit of a challenging task to comment in 18 minutes about a very complex document that summarizes new guidance from the EMA on the development of anti-seizure medications, or more generally, drugs for epilepsy. I have my disclosure here, an additional disclosure. I am an external expert to the European Medicine Agency, but I'm not speaking, of course, not representing in any way the agency. I'm just giving my personal comments about the guideline. Just as an introduction, a little bit of history. Guidelines for epilepsy from the European Medicines Agencies originated through this process between 1998 and eventually they came into operation in 2001. That was the first version. There was a second version 10 years later that came into operation in 2010. The procedure was the same for both versions, meaning the Efficacy Working Party developed a draft. The draft was put in the web. Comments were solicited from stakeholders, and eventually the document was revised based on those comments and approved by the relevant authority. A similar process applies to what we'll be discussing today, which is revision 3. It took much longer because it started in 2018 and we are in 2024, so a very long process, 2 phases of public consultations, different drafts produced along the way, and one of the reasons for this was the impact of COVID during that period that kind of paralyzed the agency for a while. So this is what you can find on the web. The latest version that is available for review is already past the phase of public consultation. Dates the 30th of October 2023. There have been subsequent changes made into the document. And what I want to emphasize, this is still a draft. So what is currently in place is the 2010 version of the guidance. But industry and in general, all stakeholders can start to look at what we'll be discussing today as an indication to the direction where we are going and more or less what we will be doing in the future. So if you will summarize this very, very briefly, there are 4 basic elements, I would say, in the revision, in the current revision. One, I would say, very welcome one is the incorporation of the current ILE classification of seizures and epilepsy. Of course, you are all familiar with the 2017 classification, and also some addition of text on study design. The second major change is new guidance concerning how the monotherapy indication is going to be approved. Then there are new sections, which were not existing before, on the trial designs for neonates and status epilepticus, and also more detailed guidance on drug development for pediatric indications. For reasons of time, I can just highlight some of these points, but cannot expand in detail. However, what I've done in each slide is put at the end the relevant part of the document, and you can go back, if you are interested to know more about a specific topic, go back to that specific reference. So let me start with the add-on designs, which is the traditional way for decades. We've been approved medications for epilepsy. This is the design you should be very, very familiar with, meaning you have an initial 8-week baseline, in patients with uncontrolled seizures on up to 3 anti-seizure medications. Then there is a randomization phase and an initial, which is optional, a titration phase, typically up to 6 weeks, but can be variable, and then a 12-week fixed dose maintenance phase. This is double-blind, and then eventually patients can go on into an open-label extension. Unlike the FDA, the EMA requires the evaluation to be done versus baseline, of course comparing placebo versus the investigational medication. The EMA requires the evaluation to be done with respect to the 12-week maintenance period. They are interested in the effect measured during the maintenance phase. This differs from the FDA that requires the evaluation to be done over the entire treatment period, meaning titration plus maintenance phase. And the new document, this difference with respect to the US position is still there. So they still insist on the evaluation being done during the maintenance phase. And there are arguments, pros and cons. But at the end of the day, the way in practice EMA looks at the data does take into account a number of caveats that comes with excluding the initial titration phase. There's been no change in the new guidance with respect to the key primary and secondary efficacy endpoint. The key primary endpoint is the 50% responder rate. Again, differently from the US, which requires a primary endpoint percentage change in seizure frequency versus baseline, so a dichotomous variable for the EU compared to a continuous variable for the US. Again, this is something which would be worth discussion, but this is not going to change. What is going to change is not a major change, but they put emphasis on some additional secondary endpoints. They want to have more information on seizure-free. More importantly, to me at least, we've been looking very carefully of these documents over time, is this reference to cumulative distribution of percent reduction in seizure frequency. I'll show you an example what that means in a second. And also they require, he said, should be assessed, so this is very strong wording, information on functional outcomes and quality of life, which were simply not there in the previous version. And have overall a broad assessment, comprehensive assessment of clinical benefit, which I think overall is is a welcome development. This is also a new part, although, if you will, they've been doing that in the past, but this is now in print, that when you're dealing with epilepsy syndrome with different seizure types, you can put emphasis with respect to the target of the trial, the purpose of the trial, on the most disabling seizure type. It's acceptable for the approval of the drug that the most disabling seizure type is reduced in frequency, even when other seizure types may even paradoxically increase in frequency or severity. This is what I was referring to. I think this is a very technical thing. We are not used as clinicians to look at data this way. The FDA has been requiring explicitly this way of representing data. It's extremely informative. And the EMA now, for the first time, requires this also explicitly, though in fact they've been unofficially requesting this for a number of years. Basically, by representing the results of a clinical trial with respect to seizure frequency in this way, you really have a lot of information. I mean, you can work down here, I don't know if you can see the arrow, the percentage change, the percentage responder rate, which is up here. This is a placebo, this is the drug, in this case cannabidiol as an example, but you can look at 75 responder rate, you can 25% responder rate, you can look at percentage of patients who have seizure aggravation. And you can look, this is a nice example of cannabidiol. This is taken from the FDA report. Yeah. EMA has no information about this, at least in the public domain. And you can see that, for example, with respect to seizure aggravation, there is a big difference between patients who are not taking clobazam concurrently to cannabidiol in the clinical trials compared to patients who take cannabidiol. Without expanding on this, but I want to emphasize, this is really a very important, informative way of presenting the results of the trial. And I guess we will be increasingly, and this is a welcome development, looking at this way of representing data. Another important point, and I just put a lot of words, and I apologize for this into my slide, but I just come back to this point, is that they explicitly say, this is some rewarding, somewhat it was there, but I think for us as a community, we should insist on this point when we deal with other stakeholders. It's acceptable to look at a specific seizure type across different syndromes. And this has been already done, if you will, in studies for the effect of a drug on generalized tonic-clonic seizures, where you're looking at patients, for example, with juvenile myoclonic epilepsy, but also juvenile absence epilepsies who have or generalized tonic-clonic seizures alone, etc. But also they quote, for example, Lennox-Gastaut syndrome. So there is nothing preventing us on the new guidance, and actually even, you may say, something encouraging us to look, for example, of certain type of convulsive seizures across a broad range of rare syndromes. And this would be the only way by which you could have a drug actually approved for a broad range of very, very rare syndromes, where it's most unlikely that we will have actually specific trials done in that condition. And I stop here in making my comments. A lot of interesting information, I come back to this, is the time to event design. which was already mentioned in the previous guideline, but there is much more extended information about this in new guidelines. Basically, what they say is because I don't know if you're familiar with this, because with the time to event, patients exit the trial when a certain endpoint is reached. They say patients on placebo exit the trial very early, which is an advantage because they are protected from having more seizures when they are receiving an ineffective treatment. But then for assessment of safety, you no longer have a control group, because as you continue the observation, the placebo group is no longer there, or has become very small. And for them, this is a problem. However, you may be aware, because some publications have already coming down in that direction, that analysis from data from previous trials have shown this really is not so much a concern if you look and analyze critically the information. But this is something where I think we can have a dialogue with the regulations. They correctly insist on accounting for the effects of drug interactions when assessing efficacy and safety of a drug. And again, this is not new, but I put it new because there is much more emphasis on this, which of course is very important. Think about stiripentol or about even cannabidiol when it was given with clobazam, and the importance of evaluating the effect by taking interactions into account. A lot of new important information concerning approval for monotherapy. And basically, the EMA recognizes that the traditional model they've been requesting, and that is non-inferiority trials in newly diagnosed epilepsy, has a major problem with assay sensitivity. So without spending a lot of time, what I put down in red here in the paragraph below is of extreme importance, because what they say on a case-by-case basis, of course you have to argue for this, it may be justified that a monotherapy trial is not necessary to support the monotherapy indication. And there are different ways by which you can look at it, but the way I personally interpret all what is written in the document, and here is just a part, is that I expect that in the future, non-inferiority trials with the traditional design will no longer be needed. And actually what will happen is, in the majority of the situations, once you have evidence of efficacy from an add-on trial, automatically the agency will approve the drug for any patients, either for use as adjunctive therapy or for use as monotherapy. So this is really a major step forward, if you will, which improve access to medications for all people, and also somewhat harmonizes the approach of the EMA with the approach of the FDA, which has been moving in that direction. Having said that, I'm personally a little bit more critical, because although they emphasize the need to exclude that the efficacy of the safety was a consequence of drug interactions, my concern is that we might end up in a situation where, in fact, the efficacy of the confounding effects of drug interactions are underestimated. But, I mean, this is a personal concern, but it's certainly a bit of a danger that I don't think, for example, personally, that the data with cannabidiol, when given together with clobazam, was enough for the FDA to justify, as they did, the approval of cannabidiol for monotherapy or adjunctive therapy. I don't think they had the evidence to do that. EMA did not do that, but based on this, they will not do that either. But the wording is such there may be an opening there. I think I'll skip this for reasons of time. Can you tell me when there is 3 or 4 minutes left, and I'll stop. So thank you very much. I didn't look at my watch when I started. And they make some suggestions about what kind of patients will be required if extrapolation from add-on therapy to monotherapy in their judgment cannot be done. And basically they revert back to the non-inferiority design. They make some suggestions which in my view are questionable. But there is not much time to expand on this, but go back to section 6.3.2, and the kind of patients that they suggest from the studies may raise some ethical questions as well. I think we can skip this as well and just say a few words about pediatric studies. They put a lot of emphasis on initially, when you start developing a trial in pediatrics, the use of exploratory strategies. So basically what they say is that if you initially start the evaluation of a medicine across a variety of syndromes, the efficacy signals are consistent across signals, then you may eventually get approval of that medication without focusing so much on specific syndromic types. This is the way you may read it. This is a very tricky, ambiguous part of the wording. But when they say, when the result of such a trial show no consistent signals across multiple syndromes, then you have to go down and look more specifically at the specific syndrome. I personally don't feel so comfortable with the way the document is written because I think there is too much ambiguity there, and I suspect eventually companies will have to seek advice from the agency before embarking on a trial. I think I skipped this, although it was important, but I'm still aware of time. They put a lot of emphasis here on what kind of designs we can apply to children that would minimize exposure to a potentially ineffective treatment. So what they are basically suggesting, and here I'm giving a summary of stacking wording, is that if you have evidence of efficacy from adults that the drug works against a given seizure type, for focal, you can extrapolate it down to 2 years without doing any study. You may do the same when the underlying etiology is the same, and they cite it as an example, generalized tonic-clonic seizures, idiopathic generalized epilepsies. If there are different seizure types, you may be required to do a specific controlled study in a pediatric population, but not a traditional approach if you have already evidence of some efficacy, even in another seizure types in adults, but you can do a shorter trial, such, for example, a time-to-event design. And meaning that if the drug already works, for example, in focal seizures, and you want to show that it works against myoclonic seizures, okay, you do a time-to-event design in which you have to decide how many seizure-free days they will have to have to remain in the study, etc. They don't go into much detail here. Specifically, maybe if I can go back there, what I note here, this is only 2 lines, but again, very important, in red. Not in red in the document, I put those in red. The study design with the time-to-event approach with variable exposure to treatment is acceptable. I mean, in those words to me, is the EMA is saying what the ILE, what the ILE has been suggesting in this document that you may be aware of, where basically using as an endpoint a time-to-event, which is the number of seizures that occurred during an initial baseline period, the duration of which is adjusted for the seizure frequency of a patient. For example, if a patient has 1 seizure per day or more, you don't need a long baseline period. You can just do a 7-day duration of baseline and then move on during the treatment with the time to event, which will be the time up to when the patient will have the same number of seizures that occurred during the baseline. And that, of course, will allow you to differentiate the drug from placebo. I'm going very fast. I hope I'm not confusing people with that. This is interesting to me. As a scientist, I'm not comfortable at all with this. And we'll have more discussion, I think, on Tuesday when we will have a session discussing the seizure modification and what do we mean by seizure modification. But what basically the EMA says, in case of an effect of a disease-modifying effect is claimed, it should be shown that the effect on seizure translates into improved neuromotor development. So in other words, if you have an epileptic encephalopathy and you control the seizures and then you get an improved cognitive outcome, for EMA, this is enough to claim a disease-modifying effect. For me, this is not science because this is still an anti-seizure medication. Stop the medication, and the cognitive improvement may well be due simply to the fact that you stop the seizure activity, which are producing cognitive deterioration, etc. To me, is this disease modification is evidence of an additional effect beyond the one that you achieve with reducing seizure frequency or controlling the seizures. I suspect at the end of the day, knowing how the agency behaves, at the end of the day, you will get an approval, not with the wording disease-modifying agent, but with the wording this medication reduces seizures and improves cognitive outcome over a certain modes of use in a certain time in a certain population. Meaning EMA will simply label what has been observed in the trial. I don't think we put a step disease modifying in the label, but this is again for discussion. And by the way, they cite also the possibility of using as controls historical controls, which is again very important, and this is new. And I'm sure you are aware that, and within EpiCARE very much so, data are being collected for rare conditions, registries to see the natural cause of the disease, and those data are going to be very useful because it will allow the approval for, I mentioned before, the extrapolation from adults to pediatric age. 1 minute? 1 minute. Okay, fair enough. Less than 1 minute. Maximum. Oh, no, I can stop right away. I already mentioned the extrapolation, so I don't need to go over that. This is my last slide, actually. It's trials in neonates. There is a fairly long series of paragraphs about this in the document. I think the most interesting part, to me at least, is the emphasis on using video EEG recording for at least 24 hours as a primary endpoint, as a way to obtain the primary endpoint for the study, which would be to eventually observe improvement in seizure frequency during that time. Primary endpoint, predefined reduction in seizure frequency. And then they want also longer-term follow-up in these newborns, but they say up to 30 days, fine, to submit a dossier for registration, but if you want to go beyond that, you have to commit post-marketing to do longer-term follow-up. So I don't think I could have done it faster, and thank you for listening to me. I'm happy to address any questions.
Thank you so much, Emilio, for this very interesting talk. We have time just for 1 or 2 very short questions. If you have questions, please come to the mic so we can record. Yes, we don't have enough space for everyone. Okay.
Okay.
Do you have any question?
If I overwhelmed you, I'm more than happy to be approached at the end of the meeting and answer any questions on a personal level. Thank you.
Levan.
Yeah. Levan?
Thank you.
The session is recorded, so we, yeah.
Emilio, what is your personal view on the use of video EEG or EEG in trials in young children? We are all against it, but it's still on paper. I mean, it's still required as I read it. So what do you think?
I also have reservations. It's extremely costly, and as you know, I don't have to tell you, it's challenging because you may easily miss an effective drug by using such a tool. I think this is an area where we can engage more with the regulators. What is fairly evident throughout the document is there is a lot of ambiguity in the word, and there is room for discussion of everything. And I suspect what companies will do. When you do a very specific trial, they will go to them and seek advice and make an argument to go one way or another. And just to explain to everyone who is not in the technical area, guidance are general recommendation, are not binding. Means companies, industry can choose alternative trial designs and approaches, and that's fine. But then they have to justify it. And generally, it's advisable, as discussed before, of course.
Since it is already a draft, it could be changed over time?
Yes, it can be changed.
Okay.
Correct. But some changes will have to be made because there are some incredible typos that need to be fixed. There is, for example, a requirement of a 1 month follow-up where I suspect they meant 12 months, etc. So I think some things will have to be, and some input has been provided also by the European Federation of the Pharmaceutical Industry. They have provided fairly extensive feedback on this. But overall, I personally think it is an improvement. I think there is too much ambiguity in some areas, but that may be perhaps something which is good to have.
Do we expect a final document?
I don't know. I asked that question to Stefan, who has been trying to get some information, and I think any time now, I suspect. Thank you.
Thank you.
Now it's my great pleasure to introduce the next speaker, Dr. Floor Jansen, the head of the Department of Pediatric Neurology in the University Medical Center in Utrecht, Netherlands. Floor is an experienced principal investigator in many sponsored and non-commercial clinical trials, including the one in which I was privileged to cooperate with the EpiStop trial. And Floor has also published some important papers on the innovative designs in clinical trials in epileptology, including N-of-1 trial. Floor will give us a talk about the new possibilities in the designs of the trials in epilepsy.
Thank you, Kasia. Thank you very much for your kind introduction. And I'm really happy here to be invited to talk to you about improvements in clinical trial and rare epilepsies, with possibilities in designs and in outcomes. And I realize after hearing Emilio speak that there's some overlap, but since Emilio had to go fast, maybe the rehearsal helps to restore your memory of all that has been said. So these are my disclosures. I will briefly mention the challenges in clinical trials in childhood epilepsy, and as I said, discuss some improvements or possibilities in designs, and also discuss some alternative outcomes beyond seizures. When we talk about clinical trials, this is just a list that is not endless, it's much longer, but there are many, many different types of trials, and we choose our trial that we want to perform based on the population, on the disease characteristics, on the intervention characteristics, and on the aims and the outcomes. But when we discuss the improvements in trial, actually mostly we think of the double-blind, placebo-controlled add-on RCT. As Emilio already mentioned, this was the main scheme that is being used, a baseline period, a titration period, and a maintenance period, and then a longer period. And when we talk about refractory epilepsy, we often talk about focal epilepsies, both children and adults, or the developmental and epileptic encephalopathies. But for a drug to be studied, we need larger populations with this design, so often in the Dravet syndrome, Lennox-Gastaut syndrome, but not in the really, really rare epilepsies. And as mentioned, the primary outcomes are usually the responder rate with the percentage change, or the percentage change in countable seizures. And this is needed for the design for marketing license perspective, but it not always reflects the clinical relevance for the patients, and therefore we need to look beyond seizures. So what are the challenges in the rare epilepsies, especially in the pediatric epilepsies? Not only the funding and the costs, which were highly difficult, but also we have to deal with parents or caregivers who act as a role keeper, so it may be more different to enrollment. They may have a personal preference, or they fear the exposure to placebo. We have the ethical concerns, again, the exposure to placebo or the withholding or delaying of possible effective treatments. Also, the time to intractability in children with developmental and epileptic encephalopathies can be only 2 weeks, really fast, because, well, they can have multiple seizures per day, and we know very soon when they won't respond to the seizure medication. Much more in the rare epileptic encephalopathies, we face the disease modification instead of symptom reduction, so we want to target the underlying condition. and it only reduced the seizures. And for that, we need to have reliable outcome measures. But with the reliable outcome measures here, we rely on the seizure reporting of multiple caregivers during the day, not only of the parents, but they also go to daycare or to school. So also these people have to report their seizures. And eventually when the trial is longer, especially in disease modification drugs, there can be reporting fatigue. If we look at other outcomes, there can be retest effects that we have to consider. In children, we often see larger placebo responses, and there's a huge variability in syndromes and etiologies with a large variability in treatment responses. So we have to better reflect the needs. Emilio mentioned already that we cannot look only at the specific epilepsy syndromes, but also at the specific seizure types, and we can look at the individuals, and I will come back to that later. What are possibilities to shorten the duration and to overcome the safety issues or the placebo exposure? And what can we do about analysis? Something has already been mentioned by Emilio. So first is the time to end seizure analysis. That was with the variable baseline duration. We can have individual number of the end seizure and also patient can be enrolled with a lower seizure burden, and they remain until the maintenance phase until the end seizure, and if it's not the end seizure it can be up to 12 weeks. And the efficacy is assessed by the number of days to the end seizure between the baseline and the maintenance period. And this can lead to a higher recruitment and also to, as Emilio mentioned, a shorter placebo exposure. Although we have to aware what has already been mentioned, that the placebo group can have a shorter exposure, and therefore it may be more difficult to assess the long-term tolerability and treatment response. The end seizure can also be the time to event analysis. It can be the end seizure, but it can also be a doubling of the monthly seizure frequency or seizure clusters that we can look at, or new seizure types, or status epilepticus. And this is a simulation of a group of patients with the seizure frequency in patients where we have the pre-randomization seizure frequency, and when we look at the 100% curve, there was no change in the maintenance period. When there was a 200% curve, there is a doubling of the seizure frequency, and you see that the patient remain in the trial shorter if we look at the red curve. And if we look at the 50% curve, which is a half of the seizure frequency, they remain in the trial longer. So you can decrease the exposure to ineffective treatments, limit the duration of placebo exposure, and may demonstrate efficacy sooner with this analysis. If we look at responder rates or change in seizure frequency, can be of interest, but actually if you talk to patients, the seizure-free days are more related to the quality of lives. So we want to look also at the seizure-free days, which is usually not the primary seizure outcome, but secondary seizure outcome. And this was an interesting post-hoc analysis of two CBD RCT studies that looked at drop attacks in Lennox-Gastaut syndrome. And they saw that the change in the number of seizure-free days differed significantly between placebo and the groups treated with Epidiolex. So this may be something that we want to include more often. The disease-modifying therapies, especially in the genetic epilepsies, has been mentioned, because we want to learn more about the disease modification. We need to have longer exposure, because we also want to look at development, behavior, sleep, all the other comorbidities that are associated to the underlying condition. And we also have to have then additional outcome priorities. One is the trial design that can capture this is the historical control trial, which Emilio already mentioned. But another option would be the step wedge trial, where you can have different groups, and you can have, with the randomization, it is decided when a certain group receives the active intervention, and it's in a different timing for each of the groups. And that can lead to both an in-group and a between-group comparison, and can capture the course of the disease, and therefore better assess the modifying effects of the treatment. When we learn about change in seizure frequency or or responder rate, we may also be interested to know how many patients should I treat to have this effect. And therefore it may be interesting to look at the number needed to treat. And this was done in this study, looking at 2 phase 3 studies for adjunctive fenfluramine in Dravet patients, and the number needed to treat to achieve a 50 or 75% seizure reduction was calculated by 1 divided by the experimental responder rate times the control responder rate. And they showed in this study that for every 2 to 3 patients with Dravet syndrome treated with fenfluramine, 1 patient achieved this 50 to 75% reduction. So this may help you in your clinical practice, I think, to think, is this clinically meaningful? Should I try this treatment in the patient? So I talked about some analysis and designs, but there are also some other endpoints that we can think of, because we really want to learn what we measure, and if what we measure is clinically relevant. So these are, this is a list that is also not endless. There can be many more endpoints, and I want to address a few examples that can be helpful, but also some where we can need to be cautious. So much is being said nowadays about patient-centered or patient-reported outcomes that actually tell us what reflects the patient perspective of their health, symptoms, and the quality of life. And they should be involved in the early phases of developing a trial, because it helps to calculate your power, but it also helps to align with the regulator interest in a patient perspective. And one of an example can be a disease-specific outcome. For example, this is a study that developed and validated a TSC PROM, where they had 82 self-reports and 75 proxy reports of items on 4 subskills: physical functioning, mental function, activities, and participation. And these can be helpful looking at intervention in this specific patient population. And also of much interest, especially for the individual patient, is the goal attainment scaling that was used already for a very long time in rehabilitation, where a goal could be to walk the stairs after the intervention. And then you have a 5-point scale, -2 to +2, where 0 is the expected level after the treatment, -2 is the initial pre-treatment level, and +2 is much better than the outcome. And instead of with rehabilitation on physical functioning, it can also be on cognitive functioning, on interpersonal relationships, on behavior, on community involvement, which can matter very much to individual patients. Thinking of disease modification, we often think of development and cognition, but we have to be cautious here, because while these 2 trials looked into developmental outcomes, the cognition was studied in the Rescue EASES trial of my group, where we assessed cognition, a cognitive index, and IQ after 6 months of treatment, and compared clobazam and steroids. And although we did find that 5 patients in the corticosteroids group increased with 11% IQ, only 5 in the group of 43 patients that were enrolled in the trial is very low. And it can be that we do not necessarily expect an increase in IQ, but we look at the progressive disease and we want to prevent progression. So is IQ then the right measurement? Also, this study, the PREVENT trial, that looked at outcomes at 2 years of age and compared pre-symptomatic with standard treatment with vigabatrin, so before clinical seizures and at clinical seizures, and they did not find a difference at the age of 2 years. But maybe 2 years is a too early age to look at differences in development. And also there was difference between the group that got standard treatment had a higher proportion of patients with TSC1. So, and we know that that is related to a milder seizure type. So the challenge with assessment of IQ or cognition is the age, the duration necessary to see an IQ change. It's difficult to capture change in an individual developmental trajectory. So we need to find better markers for cognition. Also, it was already mentioned EEG. These are 3 examples that looked at EEG as an outcome measure as well. The EpiCARE trial, and it is maybe important in those where the countable seizures are much less clear. So, for example, in infantile spasms, in absence seizures, or in patients with electrical status epilepticus sleep, now called EE-SWAS. Where it is where seizures are less countable, you can look at the EEG. And in the EpiCARE study, they did find a significant difference between the vigabatrin hormonal therapy in combination in comparison with hormonal therapy only, and also in the juvenile of the childhood absence trial with etosuximide, lamotrigine, and valproate, they did find that the EEG seizures only were different in the 3 groups. But with the rescue ESAS trial, we did not find a difference. And this study highlights that when we look at EEG, we have to be careful because there can be high inter-reader variability and low inter-reader reliability of the interpretation of the seizure types, but what we found in the EpiStop trial as well, also on the baseline EEG measurements. And also what has been mentioned, this is very costly. 1 minute, okay, but in 1 minute I will try to mention the N01 trials because these can really look into patient-reported or patient-centered outcome measures. They can determine the treatment response on an individual level, the efficacy both as the tolerability. It can be in an individual patient, double-blind, randomized, and we look at multiple crossovers in one patient with standard measures of effect. And this is particularly suitable for chronic and stable conditions with rapid onset and cessation of treatment action, rather symptomatic than curative treatments. And this is a graph where you can see that one patient undergoes multiple cycles with both the active intervention and the control period. And before the trial, you tailor the design on the individual patient, the disease characteristic and the intervention, and you predefine what would be the efficacy levels for the individual patient. And at the end of the trial, you have a shared decision-making whether to continue the treatment or not. This is a review done by Victoria de Philippe, where she only found 6 N01 trials nowadays with multiple crossovers in epilepsy, so very few, and we're developing, she addresses all the methodology used in these trials. Also, my final point is that the N01 trials can also be at an interface of research and care, where in clinical N01 trials, it is aimed to improve the clinical care for the individual patient, and the research N01 trials, they aim to answer more scientific questions beyond the individual, and they can create generalizable medical knowledge. And here, it's very important that you align with your competent authorities for this. So to conclude, we need adaptive trials for epilepsy, especially in children, or in the rare and complex epilepsies with shorter exposure to placebo, and trials that address the disease modification. Careful consideration of the design aspects can offer more reliable and clinically relevant results. In rare population, we may benefit from international collaborations. The N01 trial design offers an alternative for a trial and error approach to assess therapy effectiveness in care and research for the individual. And the patient-centered outcome endpoints better reflect the needs for the individual patients. And to achieve all this, we have to partner, first of all, with patients, but also with their regulatory agencies and competent authorities. I would like to thank my team in Utrecht working on clinical trials and N01 trials and endpoints. And I would like my co-leaders of the working group, Clinical Trials in EpiCARE, and would like to thank you for your attention.
Thank you, Floor. Again, we have time for 1 or 2 very short questions. Actually, I have one. Do you think, Floor, that with N of 1 or N of few design, we can reduce the burden of non-commercial clinical trials associated with lack of funding? Because we could have funding from different agencies in different countries and then try to combine it together or pull it together.
Yeah, well, hopefully we can do this, but also the N01 trials are timely and costly, Especially if you want to do it in a placebo-controlled manner, but it's not necessary. You can also, if you want to look at the efficacy of an anti-seizure medication, for example, seizure medication, where you think, well, the benefit-risk balance is a little bit doubtful, you can also want to look at a single open-label trial for your patients, which may also be very important. But if you look at reimbursements or registrations, then yeah, we have to await that still.
Thank you. Thank you very much.
Thank you. And our session continues with a presentation held by Professor Alexis Radzimanoglu, pediatric neurologist and epileptologist, director of the Epilepsy and Neurophysiology Unit at the San Juan de Déu in Barcelona, but most important, our beloved coordinator of a European Reference Network, ERN EpiCARE, and with Professor Eugen Trinka, director of the European Consortium for Epilepsy Trial. And actually, Professor Radzimanoglu's today's presentation is the added value of the European Consortium for Epilepsy Trials. Thank you.
Thank you very much. Okay. So, thank you very much for being here, and I would like, on behalf of the whole team, to discuss this added value of the European Consortium for Epilepsy Trials. Very briefly, and in addition to the introductory slide from Kasia, I would like, for those that do not know, although you should by now, what are the 24 European reference networks, because in a way, although it pre-existed, if we want to be honest, the European Consortium for Epilepsy Trials is a product of this process. So, as I said at several occasions during this meeting, this is an initiative of the European Commission initially, in the sense that the funding for the management comes from the European Commission, and as you can see, it covers different areas of disease. What is more interesting is that in 5 years' time, this is what they represent today. 382 hospitals, 1,619 medical teams, more than 20,000 medical doctors, nurses, fellows, and staff. In other words, I could stop here. If we want to continue doing the design of the clinical trials without the input of these people, we can do it. But this is probably not the good way, because we are going against a wall, and the wall will be presented by Lieven a bit later when we discuss precision medicine, targeted therapies, and so on. And just to make it clear why from EpiCare to ESSET, because the ERNs are not legal entities and they will never be. They are networks representing hospitals in their respective member states in a continent where we are not in the United States. It is not one country, it is at least 27, plus all the others of the European region, which means different legislations, different rules for trials, different consensus agreements, different data sharing agreements, and so on. So it is a very complex procedure, but we still need the drugs for our patients. We still need new medications, new trial designs, and so on. So what we did is, yes, EpiCare today has a very large representation. It's more than 60 medical teams that, with the support of a management team, have a regular contact between them at several levels, and I will show you in a minute how this is structured. And it is probably, this is the strongest asset of a legal entity like ESSET, because it has the input of all these people behind it. So what I am presenting is not me, it is hundreds of people behind that do the job. How did we do it? Well, we did a structure for EpiCare, again, that corresponds to the epilepsy world, which means multidisciplinary. We could not, if you look at the other ERNs, they are structured per rare diseases, anemias, blood disorders, and so on. We could not do this. And we could not do it because you have a rare and complex epilepsy, as I said, once someone made the diagnosis that you have a rare and complex epilepsy. Until then, you have epileptic seizures that can be rare or not rare, that you can go into a trial and really modify the course of a patient because he was treated earlier. But if you don't make the diagnosis, the patient still has epileptic seizures. So we structured the network to increase our knowledge at the different multidisciplinary fields. This is about all the tools we are all using in our everyday practice, but also because the ERNs have a public health mission to be the backbone of the national networks, to be able to connect with smaller hospitals in the regions, to be able tomorrow not to have an exponential increase of number of centers who do trials without having the backup and the competence to select the good patients, and I will come back to it in a minute. Well, an ERN has, and not only ours, has a public health policy mission also. If you want more details about what they are doing, one, you can visit the booth. Two, if you have an iPhone, you can QR code which is there, and you have a booklet with all the details. So I will not go more in what EpiCARE is doing. I was just trying to tell you that the existence of this network supported the launching of the European Consortium of Epilepsy Trials. I said it pre-existed. Yes, it pre-existed as a concept with Epilepsy Alliance Europe from the ELAE. The development stopped mainly because of COVID and all the other activities. And it was the fact that with the creation of an ERN like EpiCARE, we do have a working group which is the driving force of the European Consortium for Epilepsy Trials. And this is just one example of the working groups I showed a minute ago. Well, as you can see, all these people working in different fields of the epilepsies, with all the rare diseases, come up with new ideas that needed to be offered, proposed to our industry partners, to academia for next trials. So of course, a structure like the ERN already produced some ideas about the priorities in epilepsy research, and it's not by chance that when we discussed this within EpiCARE, the need of innovative trial designs and their outcome measures was included in the 6 main. It was not necessarily the first. I put it first on the list because it was the topic, but it was one of the 6 topics concerning all types of epilepsy research or fostering research in the epilepsy world. Now, and we are going more to the concrete aspect of these things. Why? Why? Because, okay, today the annual number of new patients with epilepsy in Europe is estimated to be more than 30,000 children and adolescents, 96,000 adults, and we should not forget the elderly, that I am now sufficiently elder to remember that at some years ago, there were several trials about the elderly. Then they were completely forgotten. They were lost in translation of the studies on developmental encephalopathies, which, strangely enough, concern only 2 of the 160 epileptic encephalopathies. And companies keep doing only the same 2, while all the others behind wait for a trial. The elderly disappeared, and this is a reality today. Now, of course, and this is a very well-known number, more than 30% of those new patients will develop a drug-resistant epilepsy. We are many in this room that we tried to define this, and we said failure of 2 anti-seizure medications. I am sorry to say, but the message is still not understood both by the investigators, but above all by the industry. Patients included in the trials today have a history of 6, 7, 8, 9 anti-epileptic drugs. And the one who can tell me that the Lennox-Gastaut syndrome at 4 years old is the same as the Lennox-Gastaut syndrome at 16 years old, he would need to prove it to me. The process of the disease modifies the whole trajectory of the epilepsy, and this is again ignored with the new trials. Now, we hear it everywhere, here at the main symposia and everywhere, that epilepsies are more than seizures. But we still see, and you saw some examples when Emilio presented, a permanent confusion both from the regulatory agencies, but also by pharma, between what is a syndrome. Are we looking for a disease? Are we looking for a spectrum of diseases that have different etiologies? And all this, after so many years in clinical epileptology, is still quite confusing, both for the regulatory agencies, let alone when the results go down to the clinicians who cannot even find themselves about the terms. So there is a clear need, and the previous speaker said exactly the same thing, to one day stop talking and adapt really the clinical trials to what is today a rapidly expanding field of knowledge, including the focal seizures. That again, there was everywhere were trials for focal seizures some years ago, then they disappeared, now they start coming up again, with different profiles. Some of them have a genetic component, others are just structural. What I'm trying to say it is that this is a very complex panorama, which is different from having a trial on Parkinson's disease or having a trial on Huntington's chorea or whatever. And if we don't take this into account, we will keep turning around. Now, what happens? We often forget, for example, that I said it a minute ago, that at what age you put a patient into a trial may completely modify your results, particularly if you're talking about small number of patients. Then you have those specificities of genotype, phenotype, where even if you have a genetic identification of the patients included, you still need some expertise behind to make sure that we are talking about the same type of disorder. You know that all do not have a gain or a loss of function. It is a quite complicated process. It was discussed this morning. And all this is reflected or should be reflected at the trial designs. Same applies to how to count seizures, video EEG monitoring, a plea to stop inventing type of seizures that are not in the classification, have completely different mechanisms between them, but need to be taken into account. So, I will not prolong, because Flor also gave you some examples. There is a clear need today to readapt the design of the trials, not copying and pasting the previous ones, but including a way of thinking of how these can be designed. And I'm sure that Lieben will say a word in a minute about our next challenges in precision medicine. We will not be able to tomorrow to include patients in trials if the work done previously in terms of feasibility, of criteria of inclusion, is not seriously based on new scientific knowledge and not on copying and pasting. Today, we still have the impression that even the selection of syndromes to do a trial is more or less random. And again, you know all how attached I am to Lennox-Gastaut and Dravet syndrome and to the patients that I meet every day, but I still do not understand why for 10 years we are doing trials only on those two, while there are another 148 minimum that I wouldn't dare saying nobody cares, because I don't believe it. But I think there is a domain where even if we are talking about niches, because I can understand a disease with 10 patients or 20, how you can do a trial, there are ways of doing it, provided you really communicate with the scientific community that knows about it. So, this is why ESSET was created, in fact. The idea was to support an entity with experts that can think and develop priorities in epilepsy trials, priorities on the way to design these trials and to select the patients, and quality of the patients to be selected. And why this slide? Because this needs to take place from the very first steps of the design of a trial. Doing it at the analysis stage is too late. And all those who honestly read the results of trials even before the publication know that sometimes, even if you have one simple syndrome included in the trial, the results, when you try to see the data of the patients, is not that clean as we present it. The number of trials I have seen on typical absence epilepsy where someone put the patient had focal seizures. I'm sorry, but in my whole lifetime, I must have seen 10 that had the two types of seizures together. But these people are in the trials. So there is a problem of how they are designed, how do we select them, how do we fix the primary and secondary endpoints. The feasibility questionnaires with time became a kind of technical document produced by a CRO without a scientific background knowledge. So we spend more time answering if we had a fridge at the hospital or doing pseudo-epidemiological data, how many Lennox-Gastaut will be drug resistant in a year and a half. I mean, I don't know if you can answer the question. I am incapable. I don't know. Some of them I will control them, some will get worse. And this we see it still every day. So what did we do? Well, we created a structure that brings together all these people, and it is open to everybody else that are interested, that contribute to the production of new knowledge in the epilepsy field, but also think on what are the best designs of the trials, how we can answer the regulatory needs, which are essential. We should not forget it. These are regulatory trials. And on the other hand, the trials needed by the clinicians, the natural history studies that can be supported, and so on. So what is the structure today? Well, it's the usual suspects, I would say. One or two people that have to lead this process, but everybody is welcome to take the place if they want. Sebil Taisha, who helps us construct all this from the beginning, a number of experts that act as a scientific advisory committee and try to produce a comprehensive analysis of what's going on, but above all these centers, and they are open again. The only thing we need is confidentiality and real proof that they are willing and motivated to work on this field. And of course that they have the infrastructure in the hospital to do it. So this is much larger than EpiCARE because it includes all these people. To respect the multidisciplinarity of the epilepsies, we used the same model as for EpiCARE. We created fields of expertise, task forces that are more competent in clinical pharmacology or in this or in that, to be able to contribute to the studies. I know that this can be done by individuals in advisory boards, but I know also that the input of brainstorming, of groups working together, I would say, up to a certain degree, independently of the pharma with which they are collaborating, is much more productive sometimes. And then, of course, this brainstorming is used with the collaborations. What the consortium offers today to academia and to industry, well, again, the booth is over there and you can visit it. I don't want to be more late because I want to leave some time for my friend and his precision medicine talking. But yes, the consortium can support today either companies or medical investigators that want to launch and follow a natural history study on one of the different forms of epilepsy, to help and promote coherent ways for precision medicine trials. I am sure that there is no authority in Europe that tomorrow will allow the genetic trials, for example, to be done in every hospital in Europe. This is not possible, it's too costly, and it will never happen. So even if you get a license today, there will come a moment that the governments will support some structures that have the infrastructure to do this. So let us be proactive, create the collaborations between the medical units and the pharma, instead of randomly selecting centers that then don't have the patients, don't have the infrastructure to put them on. So the selection of sites and their expertise is essential. And again, it is not at all an elitistic approach. Believe me, those that know me know how open EpiCARE is to everything. There is one point. Expertise doesn't come from the sky. You need to see a lot of patients with developmental and epileptic encephalopathies to be able to diagnose early one, to put him in a trial, to know how to explain this to the patients. So those who want to hear the message, it is not that we want to exclude one center or the other. We are as much inclusive as possible. But this needs training, this needs knowledge. Otherwise, those that go into a trial are not the good patients. So we are already in contact with many. I particularly wish to stress and underscore, and at the same occasion thank, the study consortium in the US. Jackie French supported this procedure and her group from the beginning. Eugen Trinka and myself are also members of the US Epilepsy Study Consortium, which has a much longer history than ESET. And we are working together because there are differences in how the European trials should be designed. Not for all of them, but there are. And also I want, of course, to thank all those companies that are already in contact or in contract. This is, of course, confidential information, but at least I can say with whom we discuss and what we do. This is what we succeeded to do despite the COVID, the last year and a half. As you can see, it is quite large, from natural history studies to protocol design, to selection of sites, or even to simple surveys. And if you want to have, again, more information, you just have to go and see Sybille down the corridor. She knows much better than me what we are exactly doing because she's doing the work. Thank you very much.
Thank you, Alexis.
Thank you.
Is there any question?
To myself, no.
No. From the audience?
At least not openly.
And just to introduce, if there are other questions, an important topic, ESET can help us design clinical trials, but doesn't help us to find the funds for the trials, right?
No.
No.
No, I mean, yes and no. Yes and no. I will tell you. The truth is in the middle. I suppose you're talking about academic trials, okay? Yes, ESET is not a funding agency and doesn't have the funds to support an academic trial directly. But what we do have and what we do, again, together with the work done in EpiCARE, we are following very closely all the calls from the European Union, all the Horizon calls. We can put together small and big teams, so the feasibility part is much more coherent than saying, I alone, I will study everything. And so from that point of view, yes, it helps. And the other way is that when you want to do a natural history study or you want to do an investigator study and you want to do it on a specific drug, the fact that we can build together the project with the consortium and with other centers, we hope if the pharma gets the message that they can help you and your trial even better than if you presented yourself alone, hoping that they will do it. But of course, today, we do not have a bank that can give you a loan, if that's the question.
But finally, even if you have an embryonal idea, please refer to Sybille because she can also help to try to find funds.
Yeah,
right?
That's why I told her to have 2 email addresses so I can still control it.
Thank
you very much.
Now it's my turn and pleasure to introduce our last speaker, who is Professor Lieven Lahe from the Department of Pediatric Neurology in the University of Leuven in Belgium. Among Lieven's numerous achievements, I wanted to mention one, which I could call service rather, which was his long-lasting past outstanding service as a chief editor of European Journal of Pediatric Neurology. And Lieven will give us a talk about the repurposing trials and precision medicine. Thank you.
Thank you, Kasia. Thank you, Valentina. Thank you, Alexis, for giving me some minutes to give my talk. So if I can find my slides. Yeah.
No, it doesn't work. Sorry. Okay.
Good. I would like to talk about the difficulties, the hurdles in the trials that we see happening in childhood epilepsy, and especially talking about repurposing and precision drugs, as has been announced by the previous speaker. So these are my disclosures, and actually this is the landscape. This is what we are seeing right now happening if you look at treatments for epilepsy or also for childhood epilepsy. And I like this schedule because you have like the 6 steps and we're moving upwards, and we have more and more precision or targeted medicine or drugs. And as you see, number 5 and number 6, or actually also number 4, are really the genetic treatments, as we call it. But before that, we have like step 1, step 2, and step 3, and I will give you some examples of all these steps. So we have, in the very recent years, seen a lot of new drugs. We have also seen a lot of repurposed drugs, and we are now very close to the very first approval of a genetic treatment, again for, I'm sorry, Alexis, Dravet syndrome. So if I summarize the ways to have a new drug, in my view, there are like 4 ways. First of all, you have intelligent serendipity, and that was the past, but also is still happening right now. Then you have the intelligent finding the pathway in epilepsy. And then what is happening right now, actually, most frequently, is a third one. You understand the functional effect of a variant or a mutation, and you try to match it with a product or with the treatment. So I call this the Tinder for genes. And then you have the Holy Grail, question mark, the Holy Grail, the genetic therapies that are really very close now. Now let's start with this first category, the drugs that were found by chance, actually. And I go back to this old, very well-known story in a potassium channel 2, KCNQ2. We all know that there's a benign phenotype, and there is a very encephalopathic phenotype. And this very first simple clinical paper in very few patients actually showed that in this KCNQ2 encephalopathy that probably sodium channel blockers were effective. And this was really not a very high standard, I'm sorry to say, a very high standard. It was a retrospective review, but it has a major impact, and actually it was the start of this thinking about gene matching. So this was proven also that the sodium channel blockers, both in the benign group or in the encephalopathic group, if you look at the effect size of sodium channel blockers, I mean, 90% become seizure-free. It's less obvious, of course, in the epileptic encephalopathy group, but still 70% of the patients are doing really okay with these sodium channel blockers. So that was serendipity, actually, because at that time, nobody understood why a potassium channel would work in why a sodium channel blocker would work in a potassium abnormality. Now we know, of course, much better. Then the other story is actually still exciting, the fenfluramine story. It's actually a very nice example of repurposing a drug, and I was quite involved in that story. I mean, it's all started when I graduated from medical school in 1986 with the Pope of childhood epilepsy, Jean Cardi. He published this paper on 3 adolescents with very specific epilepsies, photosensitive epilepsies. And now if I read back again, the first case report, actually there was a typical case of a sunflower syndrome. So these adolescents would have seizures by very specific photic stimulation. And he showed that fenfluramine, at that time, they didn't know why it would work. They didn't know it had something to do with serotonin. He treated this with high dose fenfluramine, actually. Didn't realize at that time it was high dose, and all the seizures went away. Now, the serendipity or the chances that actually in the clinical practice we realized that children with Dravet syndrome also had photosensitivity, especially in the first years of life before you start valproate. And it's only because making this link that Dravet children also had photosensitivity that we tried fenfluramine in this particular syndrome. And you all know with some success, it's now marketed for Dravet syndrome. Only after that clinical finding and only after the marketing, actually, we tried to specify the working mechanism of fenfluramine. Why would it work in Dravet syndrome? Why would it work in other infantile epilepsies? And it has something to do with the very difficult to understand sigma-1 pathway, but especially also with serotonin. And we could show in a lot of preclinical work that fenfluramine indeed works on serotonin receptors, but especially on serotonin 1D, 2A, especially on 2C and 4 and 7. It also restores the deficient arborization of a GABA interneuron if you give fenfluramine early enough in a preclinical model, which is quite unique if you look at efficacy of a drug. So actually we understood, or we tried to understand why fenfluramine would work in Dravet syndrome. We identified these receptors, these serotonin receptors, and now the next phase is, of course, that there is now a new drug almost ready for clinical trial and probably on the market in a few years, having a very selective 5-HT2C receptor, just like fenfluramine, but very selective for only this serotonin receptor, and the very initial results are very promising. You can see that like in the Dravet syndrome, in the Dravet children, there was a 60% decrease of seizure frequency compared to a small group of placebo children. So that was actually first finding a clinical drug and then finding actually the working mechanism. Pathway discovery, I mean, there's no better example than the tuberous sclerosis. I mean, we all know about the pathophysiology of tuberous sclerosis. We know there is something wrong with this pathway because of a mutation in one of these two genes. There's an overactivity of this mTOR pathway, and with mTOR inhibitors, we could modify or change that pathway. And actually, this was already known 15 years ago. It has a very good effect, these mTOR pathways, in the growth of the tumors, as we all know, in tuberous sclerosis, but also in epilepsy. And the very first paper published by the group of Paolo Curatolo, as you can see lower right, after the start of an mTOR pathway inhibitor, you could see that the seizure frequency went down. This was a single case report. So we actually are influencing this pathway very dramatically. I mean, this is very precision medicine, I would say. So we were hoping that if you give these drugs in tuberous sclerosis and you look at the epilepsy, that it would be a dramatic effect. And we all know the EXIST trial, and I keep repeating that this is a good trial, and it has a significant finding, as you all know. Also, if you look at seizure-free days, as we heard before, so if you compare placebo with the two groups, concentration group, but actually we were a little bit disappointed because it's only 50% decrease of seizure frequency, as you can see with any other drug. So did we do something special giving this drug to tuberous sclerosis? So why is it not more effective? And the reasons are probably obvious for most of you, and I think it's very true that this trial started at the age of 2. And a lot of epilepsy syndromes, I think we really need to start much earlier, and that's also a message for the genetic treatments. I think earlier treatment will give better results. And now we have some data to show that this is indeed true. You need to start earlier with these treatments in tuberous sclerosis. And I refer to a very recent paper by Kasia Sergius and the Polish group. If I understand this very well, because it was a difficult to read paper, this was a group of -2 years. They were treated with this with mTOR inhibitors, and they compared it. It was a retrospective study with standard treatment in red. So after 3 months, after 6 months, 12 months, and 24 months, they looked at the efficacy of the mTOR inhibitors that were started before the age of 2. And actually, you could see that compared to standard treatment, you see that there was a better effect. Not always statistically significant, but a better effect. But the message is clear. I think we should start earlier with this sort of drugs. Now, another personal example, actually, of a pathway discovery started in 1996 with a very short paper on 2 brothers, young children,
And they had psychomotor retardation, as it was called that time, very difficult to treat epilepsy, actually hypsarrhythmia and infantile spasms, if I read the paper again. They also have microcephaly, but they had a very low level of serin in their cerebrospinal fluid. And actually, these were 2 cases, and then there was another paper from the Italian, but so far I didn't find any other cases. These children had a problem with this pathway, and they especially had a problem with this enzyme, the phosphoglycerate dehydrogenase enzyme. And actually, they could find in fibroblasts at that time, because they didn't have a genetics, that there was a low level of this enzyme. So this is an important pathway. And actually, these children, these 2 brothers, were treated with serin, and the epilepsy stopped, and actually also the head started to grow again. So this was a unique finding, giving serin in these 2 brothers. Now, if I look to that pathway again, and I was contacted with from another lab in my university to look at these findings. Now, this is, again, this enzyme in this serin pathway, but actually, if this pathway works very well, you consume glutamate and you increase GABA. So for an hepatologist, this is like a dream world, because you increase GABA and you decrease glutamate. So the next step was, imagine that we can actually accelerate or pimp up that pathway. So let's try to find modulators or enhancers of that enzyme so that we can actually increase the level of GABA in patients or in children with epilepsy. So another lab looked in vitro whether they could find products, repurposed products, existing products that could enhance the activity of that enzyme. So more than 3,000 products were tested. This is a known library. And actually, we found several products that really could enhance. This is the standard, and this is the enhancement of the enzyme activity. And actually, the best one was cleoquinol. And cleoquinol is an old antibiotic drug for gastrointestinal infections. And that's still used in dermatology in some countries. So cleoquinol is an old drug that is not used anymore very frequently, but is one of the very strong enhancers of that drug. So the next thing was to go, that's probably a next repurposing drug for epilepsy. The next thing was to go to animal models. And we used our zebrafish. This was done in the zebrafish lab in our university. And so this is the control condition. And here you give the AKP. This is a very difficult to pronounce name of the product, but I will say AKP. You actually see that these fish start to have seizures. And why is that? Because AKP lowers GABA in the brain of these fish. So this is control, this is after AKP. But then if you add cleoquinol, CQ, you see that you actually restore to like 30 or 40% of the phenotype. So actually in zebrafish, not only looking at seizures, but also looking at the electric activity, this drug works in this model of epilepsy in a zebrafish. Next step is to go to rodents, and in this 6 Hz psychomotor seizure model, again, so this is the way it works, this is the control, and this is with increasing dosages of this drug, cleoquinol, you see that the duration of the seizure goes down. So this is a typical laboratory academic exercise to hopefully find a new drug for a child with epilepsy, based on the fact that perhaps you're doing something in that pathway. Now we are running a trial, of course, and we have like 7 children now included, and I cannot disclose the results, but some of the children are doing quite okay. Next is the most frequent one nowadays. This is this Tinder for genes. And again, I will start with a lovely example. Not doable in everybody, but this is a paper from the group of Oren Devinsky, and they had a rich family. It's very important because they funded the whole research. This was a 29-year-old woman with Lennox-Gastaut. She was actually quite well controlled with felbamate and topiramate, not too many seizures, but she still had, as usually, a lot of multifocal discharges and slow spike wave. The etiology of her Lennox-Gastaut syndrome was a mutation in one of the GABA genes. And you can see it over here. Actually, This is the control condition, and in her mutation, you don't see any current of the GABA receptor, or you have to give very high doses of GABA before you see any current. So they try to find a drug that would specifically help for that patient. And again, 1,320 components were tested with the hope that they would find a drug that would increase the GABA currents. And the best one was vinpocetine, which is actually derived from this very lovely flower. Now, if you look at the results, so this drug was given to that lady, and actually this drug is also being used in some trials for dementia and cognitive improvement without huge successes. Now, if I read the results, there was a dose-related decrease of epileptiform discharges. Remember, she was almost seizure-free, and she had an improvement of global clinical function. So this is a very NS1 trial again for this simple drug in a GABA receptor gene. Now, this was picked up by the group in Belgium, in Brussels. They identified a child with actually a child with absence epilepsy moving into a juvenile myoclonic epilepsy, but very difficult to treat, which is not the usual case. And in that particular 8-year-old boy, there was a mutation in GABA in the second or number 2 gene of GABA. So not number 3 as the previous one. And they also tried vinpocetine. And if you read the abstract, after treatment with vinpocetine, the patient showed a dramatic initial response, further reduction of seizures, and improvement of his cognitive function. But I'm a very strange one because I not only read the abstracts, I also read the rest of the paper. And if you read the rest of the paper, actually you should be a little bit more cautious, because after initial very good phase, then the child returned to normal seizure frequency levels. He was a bit better, but not actually that dramatic. So, next example of this gene matching is SCN8A, and it will show you what you will see for most of the genes, actually. This is the data from a patient registry, actually looking at what do patients like the most, what drugs are the best in SCN8A, without telling anything more about the gene abnormality. And you see that in purple, they like sodium channel blockers. They are doing the best. And to my surprise, and I still do not understand why, the patients didn't like levetiracetam for treatment of this condition. And this is just an example of things that are happening for almost all the genes, also for SCN1A, SCN2A, now for SCN8A. It really is important, and we learned this morning again and again. The next step in genetic diagnosis is to understand the functional repercussion. Is it a loss of function or a gain of function? And here you see again with the gain of function, you have many more developmental encephalopathies. So with the gain of function, of course, you should give sodium channel blockers, and here is a nice result. These are the sodium channel blockers. In the gain of functions, they are doing much better than in the loss of functions. So this is what we are learning, and a very recent example, this is the last example I could find in this gene matching thing, and it will be published in this congress also by Johannes Lemke. Very nice paper, and actually this paper shows what is happening right now in development of drugs and understanding genetic epilepsies. This is another potassium channel. I think 14 patients, most of them had epilepsy, most of them have severe multi-morbidities, as Ingrid Scheffer was saying, not comorbidities. And then they all had a mutation in this single gene. And as in the SCN2A, 8A, now also with the potassium channel, they look at the functional repercussion, and you see that in a lot of these cases there is a loss of function, but in other ones there is a gain of function. And here you see the position of the mutations in the channel, so this is very nice work. And now if they look this single patient with this gain of function mutation, they found out that one drug, fluoxetine, is really effective in restoring the activity of this channel. This doesn't mean that this drug will work for the epilepsy in this particular patient, but this is the way we are thinking now of developing new drugs. Last is Holy Grail. Thank you. So the genetic therapies. And I mean, the Congress this week is full of papers on genetic therapies. And again, I'm sorry, Alexis, but the most progress has been made in Dravet syndrome. And this is the the trial with pimping up the normal allele in the Dravet syndrome, you could increase the sodium channels actually in these patients, and the preliminary results will be presented, not the preliminary, but the next results will be presented, and we are close to actually clinical use of that drug. I think in 1 or 2 years it will come in the US and then later in Europe. And the other one is the single shot AAV, so virus-based treatment for SCN1A. Also a very intelligent design. I mean, these drugs or this therapy actually targets specifically the GABA interneurons and actually is increasing also the number of associated sodium channels in the cell membrane. So the results still have to be expected for these trials, but we are hopeful that this will happen. And actually in this last paper, and you should really read this one, this is a wonderful review on current status in epilepsy. I found these tables on the current status of the genetic treatments, and I mean, it has changed. The last 5 years, there are many trials going on, phase 1, phase 2, phase 2A, trials in many other diseases than only Dravet syndrome. But, and that's my last comment, actually, we still have to understand why we already know right now why these genetic treatments, we understand that they are not working for everybody and that the effect size is hopefully much better than an anti-epileptic drug, but it could also be that it's only just comparable to an anti-epileptic or an anti-seizure drug. So why is that? If you're so specific in targeting sodium channels, why is it that these drugs will not work for 100%? I know we cannot expect this, but why not for 80 or 90%? We will see nice results of 60 and 70% seizure frequency reduction, but why not more? And I think that's all because we still have to understand all these other modifying things that are dictating the results of a trial. And age is particularly important. I mean, you have to start early with all these treatments, especially in the DEEs, but also modifier genes. And we start to understand these modifier genes and also the other factors. And someone called this, we need to understand the personomics of a patient before we understand why things are happening or not being effective. Just to give you an example, old paper, typical Dravet children, but if you also have a VUS, a non-significant variant in the POLG gene, well, I know it's only 2 patients, they were likely to develop more severe status epilepticus. And it was confirmed in another paper. And also there was another modifier gene in GABA for Dravet syndrome. But the most interesting paper in that respect is the paper published last year in Brain. They looked at adults with Dravet syndrome, and they, with very well-known phenotypic Dravet syndrome, pathogenic SCN1A mutation, but they looked genome-wide what is also wrong with these patients. And actually, they compared it with the database of the London epilepsy patients and the non-London control patients, more than, I think, 100,000. And they compared these genomes between the Dravet adults and these control patients. And as expected, in the Dravet adults, there were many more other genes, not VUS, but other pathogenic genes that were associated with this phenotype. And actually, at the end, they could find in 4 genes variants that were really important to dictate the phenotype. So they had an SCN1A with the DEP-DCM5, SCN1A with CHD2. So I think we are getting there. I think it will be a working together of different genes in one syndrome that dictates the phenotype. But that's also the explanation why targeting only SCN1A, as in the genetic treatments, probably will not give you the answer and will not be very or 100% successful. So thank you very much for your attention.
Thank you. Thank you, Lieven. I particularly loved your combination of serendipity, Holy Grail, and the Tinder. We have time for 1 or 2 questions.
Okay, super.
Thank you.
So we finished this session. I would like to thank you so much, all our valuable speakers. And since there was not too many times for questions, I hope they are available to better respond to our question also during the Congress time. Thank you again.